like to welcome all our attendees on behalf of Dr. Jabri, myself, the Chicago Celiac Center, and the Celiac Leadership Council. We'd really like to welcome everyone. I know these are strange times where we are now on a Zoom platform where none of us can really see each other, but hopefully we can spend the next few hours together getting to know each other through our work, getting to know each other through our questions and answers. And I know that I'm sure we'll be encountering some technical issues, but as we go through that, hopefully everyone will have patience with us. So one of the things that we had wanted to do as a part of the symposium was really for everyone to get to know about celiac disease, which a lot of people do know, but not just celiac disease, but to look at where are all the other diseases that are associated with celiac disease, looking at autoimmunity and beyond. So for today, there's going to be a little bit of a clinical bend, a little bit of science, but a lot of collaboration. And I do hope that by the end of the day, everyone will see that celiac disease is sort of the entry point for a lot of other diseases and conditions. So before I introduce the moderator for our first session, just have some housekeeping things. One of that is if you have questions, please put them in the Q&A, and the moderator and the chair will be looking at the questions, and at the end of the session, all the speakers will be able to help answer those questions. You will also have received a CME information sheet. Please do fill that out for credit instructions. And one of the things we really do want is for this to be very interactive. So send in your questions. We hope we'll be able to answer some of the questions. And before we actually get started with today, there are some other people who I really would like to thank. And without them, I don't think we would have had any success. So a very, very special thank you to Mary Gibson, who's with the Individual Giving, Peggy Hassanauer, who I think without her, this program would not have happened, Brad Jutras, who's with our development team, and of course the team that has put all this together for us from a Zoom standpoint with, as you can see, Lisa, Monica, and Lindsay. The education, the getting us trained for this has been phenomenal, so thank you. And in order for all of us to get the CMEs, Tom Weber has always been our support. I leave Karen to the last, but she really is the pillar and the backbone of getting this together. The number of hours, days, weeks that she has spent sort of cajoling us, getting our talks together, sending them to her. I don't think that we would be here together today if Karen had not been pushing all of us. So without much more wastage of time here, I would like to introduce Dr. Carol Semrad. She is an esteemed member of the Celiac Center Group and also the director of the Adult Clinical Nutrition and Small Bowel Disease. So at this time, Dr. Semrad, I'm going to hand it over to you.
Thank you, Dr. Verma. Good morning, everyone. I'm pleased to be moderating this first session. And I would first like to thank our sponsors for this session, Brooke and Duncan McLean and Karen and Eric Simon. Our first speaker is Bana Jabri, who I work with at the University of Chicago Celiac Center. She is a professor in the Department of Medicine and Pediatrics and the Director of Research at our Celiac Center. And she is a leading researcher in the area of a celiac disease. Her talk will be Celiac Disease, an autoimmune disorder driven by a dietary antigen. So welcome, Bana.
Dr. Semrad, we'll actually have to start with...
Sorry, here it's Luis. So I'm actually on the phone with Bana. She's having some issues logging in. So just maybe 1 minute.
Okay.
Sure. Can we go ahead and get started perhaps with Dr. Anderson?
I think we have we've got some minutes here, right?
That's right. It was
Start
at
till 8:45. So why don't we try to set the stage with Dr. Jabri if we can.
So Dr. Semrad, perhaps we could talk a little bit about Chicago.
Yes. Let's welcome everyone.
Yes, and it's actually not a bad day today here, a little cloudy, but otherwise not as cold as people would expect it to be. And I think, from a Chicago Celiac Center standpoint, our center has definitely been growing. And Dr. Semrad also, how about telling us a little bit about the adult group that you have?
Well, I think number one, we're unique in that we have both an adult and a pediatric program. So we have continuity of care across the lifetime of a celiac patient. And we have phenomenal basic research. And when you have those two, good clinical care and phenomenal research, you have the ability to translate new discovery into practice. And I think this is about the best model we have for really pushing disease care to cure forward. So I think I'm very happy I'm in this kind of center because it makes for just a very enjoyable career. Let's put it that way.
Absolutely. And also having the collaboration, since we are talking a lot about autoimmunity and diabetes today, working with our diabetes colleagues, and you'll see all of them today as well, but having joint sessions and joint clinics is something that is, of course.
We have strong clinical autoimmunity too of rheumatology programs. So we really are lucky, fortunate.
Absolutely. And at this point, I see Dr. Jabri has joined us and I will take off.
Okay, good morning. Morning, Bana. I just introduced you already so everyone knows who you are, and I'm going to mute myself.
You are muted. Can you hear me?
Yes, we can.
Okay, so I'm starting. So first of all, thank you very much for joining us. It's wonderful to be here all together. And the reason we really wanted to put this symposium together is because we feel that celiac disease is really at the intersection of autoimmune disorder and intestinal inflammatory disorders. So my talk is really going to introduce celiac disease in that context. And as you can see in this image, there's Dr. Stefano Guandalini, who used to be the director of the Celiac Center at one of our events. So there's no specific financial interest to disclose.
So we cannot see the slides. Sorry.
You cannot see my slides? Wait a second.
I certainly cannot.
Okay.
No, I can't.
Share. Okay, so here. Do you see them now?
Yeah, we're good.
Okay. Yes.
Okay, thank you.
Yes.
Yeah, so here. Yeah, so I was just pointing out Dr. Stefano Guandalini because he's very special to us and he was the person who really founded the center. So I wanted to point him out and say how much he has done for us to put together this center. So why is celiac disease considered more like an organ-specific autoimmune disorder? So when you think about celiac disease, you can think on one hand, all the diseases that are linked to dysregulated immune responses to dietary antigens, such as food allergy. But unlike food allergy, celiac disease is generally not associated with systemic symptoms and with anaphylaxia. And on the other hand, you have inflammatory bowel disease and other intestinal inflammatory disorders, but those disorders are associated generally with the overall destruction of the tissue. What is characteristic of autoimmune disorders is that on one end, you have antibody production against self-antigens, and on the other end, you have the destruction of one very specific cell type. So instead of having a general destruction of a tissue, the immune system targets just one specific cell type. In type 1 diabetes, it is the beta islet antigen, and in celiac disease, it is the epithelium that lines the surface of the gut, and that is responsible for absorption of nutrients and vitamins. So in the context of celiac disease, what is particular is that we know the driving antigen, it's gluten. In presence of gluten, you are going to mount a very specific immune response against this driving antigen, which is gluten. In autoimmune disorders, it would be against a self-antigen. But what is particular in the context of celiac disease is that this activation of gluten-specific CD4 T cells is going to be associated with an activation on one end of the cytotoxic T cells that are going to selectively destroy the surface epithelial lining, leaving the rest of the intestine intact. And on the other end, you are going to have the generation of antibodies that target a self-antigen, which is an enzyme called tissue transglutaminase. So it has all these features of autoimmune disorders, which is that there's a very strong MHC class II association with HLA-DQ2 and DQ8. There are presence of autoantibodies, and you target just one specific cell type that is going to be destroyed. So the reason we are really very involved in the study of celiac disease is because, of course, many people who suffer of that disease have a big impact on the quality of their lives and also have many secondary symptoms. But on the other end, also celiac disease is a really unique model to understand organ-specific autoimmune disorders. For many organ-specific autoimmune disorders, people were really studying animal models because they were not able to target the tissue or analyze the tissue in human. And human studies are really key because these are the studies that are going to actually tell you what is happening in the human disease. And even if animal models have the same end outcome, it doesn't mean that those animal models reproduce faithfully the initiation event and the actual pathways that are involved in the disease development. So human studies are key. And what celiac disease offers is that we know the driving antigen, it's gluten, and this can be controlled because you can study patients who have active celiac disease that eat gluten, and you can study patients who are on a gluten-free diet. So where you have withdrawn the driving antigen, which is not possible in other organ-specific autoimmune disorders. Furthermore, as I said already, you can analyze the tissue that is targeted by the autoimmune process. And even if blood can be helpful to be analyzed, it is really, really key to understand what is happening at the level of the tissue to really understand the pathways that are involved in the development of an autoimmune disorder. Furthermore, we know that a disease doesn't happen from one day to the other. For organ-specific autoimmune disorders to develop, there are several steps that are required, and it is a step-by-step process. And it is very difficult to access, again, the patients and the tissue at these different stages of the disease. This is different in the context of celiac disease because we can have access to intestinal control tissues because people undergo endoscopies for instance, for abdominal pain and end up having a normal intestinal tissue. And also you can access the intestine of patients only when they have antibodies, autoantibodies, when they have active celiac disease, when they're on a gluten-free diet. And also a number of family members wish to undergo endoscopies when they have intestinal symptoms. So it's again a unique opportunity to study the tissue and the disease process at different stages. And furthermore, because we know the antigen gluten and we know the HLA, one was able to develop unique tools to really follow the T cells, the immune cells that are specific for the driving antigen. So when we think about celiac disease, we really think about the disease at different stages, and we know that they can be dissociated. On one end, you have the development of a dysregulated immune response to gluten. And this dysregulated immune response leads to the production of antibodies against tissue transglutaminase but this is not necessarily associated with the destruction of the intestinal lining. This would be a little bit the equivalent of LADA, latent autoimmune diabetes of the adult, where you have autoantibodies, but you don't have yet destruction of beta islet cells. Also the equivalent of the mouse model of type 1 diabetes, where you have infiltration of the beta islet cells by immune cells, but the beta islet cells are not destroyed. And this really has taught us that additional signals and other pathways are involved to lead to tissue destruction. So the 3 questions I want to discuss with you today is what makes gluten a special dietary antigen that can lead to tissue destruction? Why is it different from other food antigens? What triggers actually the induction of an inflammatory immune response against dietary antigen, and what causes tissue destruction? So through the work of many, which include Fritz Koenig, Ludwig Solid, and Bob Anderson, who is going to talk later today, we have really learned that gluten is a particular protein in that it has a lot of specific amino acids, such as proline, that make it resistant to proteolytic digestion in the gut. And why is that important? It's because to activate the immune system and T cells, you need to generate long peptide that are able to bind to what we call antigen-presenting cells, which are cells that present those peptides to the immune system. Furthermore, there is this enzyme tissue transglutaminase that can modify gluten and that can introduce a negative charge. And why is that important? Is because the predisposing HLA molecules, DQ2 and DQ8, have positively charged packets, and we all know that positive attracts negative. And so when you introduce these negative charges, you all of a sudden make these peptides of high affinity for these predisposing HLA molecules. And so what we have learned over the years is that in order to really induce pathology, you need a perfect storm. And this is really achieved in the context of celiac disease, because gluten is a key substrate for tissue transglutaminase that can modify it and make it much more affine for these predisposing HLA molecules. So what is happening is that even if in the context of celiac disease, you will generate immune responses against dietary antigens, and we know that because there are antibodies against other food antigens, the T cell responses they can induce are very minimum. They are of very low frequency. And we know that in order to get pathology, you need to achieve a threshold of the frequency of these pathogenic T cells. So if you don't acquire this number, you will not see pathogenesis. And what happens in the context of gluten, transglutaminase, and HLA is that you are able to amplify this immune response and to reach this critical threshold that then is going to mediate disease. And this is what makes gluten different from other dietary antigens. But presenting peptides to the immune system does not mean that you are going to induce an inflammatory immune response. And we know that in the context of the gut, we have evolved to induce actually a tolerogenic immune response and not an inflammatory immune response, as described in celiac disease. So the discovery that in celiac disease, you mount an immune response against gluten that is inflammatory really posed the question, what is happening in the context of the gut that now instead of inducing a regulatory immune response against gluten, you are inducing an inflammatory immune response? There were several epidemiological studies that suggested that a viral infection could play a role in the pathogenesis of celiac disease. And there are also several studies showing that type 1 interferon, which is really what is induced upon viral infection, are upregulated in the context of celiac disease. We also know when we analyze the prevalence of celiac disease and look at the frequency of predisposing HLA and wheat consumption, that there is not a perfect correlation, meaning that there must be other environmental factors that drive the development of celiac disease. And this is really exemplified by what is happening in the region of Karelia, where one part is in Finland and the other part is in Russia. And the prevalence in Finland is of 3% and in Russia is 0.3%, really indicating that other environmental factors than gluten are key for the development of the disease. We also know that the predisposing risk factors, genetic risk factors for celiac disease, have been positively selected through evolution, and Luis is certainly going to talk more about that, suggesting that the genes that predispose us for autoimmunity probably have also played a role to protect us against infection. So what we did to address this question is we established a collaboration with a virologist, Terry Dermody, who is now chairman of pediatrics at Pittsburgh. And we asked the question, can we really separate what is an infection and protective immunity and clearance from the virus from what we call immunopathology, meaning the development of a dysregulated immune response that can ultimately lead to pathology. And what we found is that if we look at reovirus, which is a very frequent infection that takes place in children, if you look at one type of virus, this virus will induce protective immunity, will be cleared, but it will not disturb the intestinal immune system and will not disturb how we respond to dietary antigen. Another virus from the same family, this time is able to change the antigen presenting cells that take up gluten peptides and present them to the immune system. And this time you have induction of an inflammatory immune response. And so what this has taught us is that we cannot just talk about viruses in general. We really need to study them in much more detail and understand what are the specific interaction between the virus and us that are going to lead to dysregulated immune responses, and that we need to dissociate protective immunity from immunopathology. And the way the gut does that is by compartmentalizing the immune response against pathogens at specific sites called Peyer's patches, while other immune responses that were against dietary antigen will take place in a different place, which is the mesenteric lymph node. And so the pathogenic virus has to disturb the antigen presenting cells that present gluten. We have studied other viruses such as norovirus, and we have confirmed that it is really the targeting of the antigen presenting cell that is going to present gluten to the immune system that is key, and that what is happening is that now all of a sudden the immune system gets mixed up and thinks that that gluten is like a virus and induces an inflammatory immune response against gluten. But what we have observed is in that case, you don't see tissue destruction, meaning that the induction of this inflammatory immune response against gluten is not sufficient to induce tissue destruction. So there has to be another event that leads to the activation of these cytotoxic T cells, these killer cells that will start destroying the intestinal epithelium. So over the years, many people have reported, and this is a characteristic of celiac disease, that there is a massive infiltration of the epithelium by these lymphocytes that are called cytotoxic T cells. And this infiltration is correlated with the development of villus atrophy. But as I said, both in mouse models, but more importantly in human, we know that this immune response, while required against gluten, is not sufficient to induce tissue destruction. So anti-gluten immunity is required, but not sufficient. The reason people were very confused for many years is because these cells that infiltrate the intestinal epithelium were not specific against gluten. So people were not understanding why they would induce destruction. And so something we proposed many years ago and wrote a perspective with Peter Green was to say that what is happening in the context of celiac disease is that the epithelium in celiac patient is going to be stressed in the presence of gluten. And this is going to send signals to the immune system that the tissue is in danger and that something needs to happen. So in order to really address that, we established a collaboration with multiple groups internationally and here, with the Mayo Group, with Joe Murray, with Peter Green, and with the group of Riccardo Troncone in Italy. And we asked, let's look at different phases of the disease. Let's look at patients that have only the adaptive immune response against gluten. And what we saw is that those cytotoxic T cells were not activated and the epithelium was normal. Then we looked at family members, and this was one of the surprises. 30% of family members, even though they looked as if they had a normal intestinal epithelium, when you started analyzing with much finer techniques such as electron microscopy and immunohistochemistry, we found that their epithelium was actually stressed. What was interesting here is that these cytotoxic T cells that were in between these epithelial cells were a little bit activated, but not sufficiently. And it was only when both events were happening at the same time that these cytotoxic T cells were becoming activated and were inducing villus atrophy. So this led us really to this model where even when cytotoxic T cells become activated in response to a foreign antigen, if they go in a tissue that is really at peace, they are not going to start destroying the tissue. And one can understand from an evolutionary perspective why that is so. It's not because a virus is present in a tissue that this virus is going to cause damage. And if it doesn't cause damage, we don't want to destroy the tissue unnecessarily. So what happens is that it's only when the cytotoxic T cells meet a tissue that is stressed and receive these additional signals that these cytotoxic T cells will become licensed to kill and will mediate tissue destruction. That also makes sense from an evolutionary point of view, because in the context of viral infection, when the viruses downregulate all the surface antigens that lead to the activation of cytotoxic T cells, the fact that cytotoxic T cells can become activated by innate signals allow them to overcome this subversion by viruses. So having found all this in human, we said, okay, now let's see if we are right, and let's retro-engineer a mouse model based on these principles. And that's what we did in collaboration with Valerie Abadi. And what we found is that indeed, if you have a mouse model that combines epithelial stress as assessed by this overexpression of interleukin-15 in the epithelium, which is really upregulated as soon as cells are stressed, and you combine it with an adaptive immune response, an inflammatory immune response against gluten, now you have a mouse model where the mice develop villus atrophy in a gluten-dependent manner, because when you withdraw gluten, the villi come back to almost normal, like in human, and you see also the presence of anti-DR-mediated gluten antibodies and transglutaminase activation, like we see in celiac disease. And importantly, we now know that this model is also HLA-DQ8 dependent, really combining all these key features of celiac disease. And so this model will enable us now to address the intricacies of all the necessary interaction between multiple cell types to understand how tissue destruction occurs. And we can also use this mouse model to test now different new therapies and also identify new checkpoints that could be used for the treatment not only of celiac disease, but also of autoimmune disorders. So, of course, I want to thank the people in the lab. I always had an amazing group of young collaborators in my lab that have really driven much of the work. A number of them are now independent investigators. Valerie Abadi is a wonderful collaborator. She has now her own research program on B cells. And then, of course, nothing could be done with a very close interaction with physicians at the University of Chicago, both pediatrician and adult gastroenterologists, and also people at the Mayo Clinic in New York and in Italy. And then also there's a very close collaboration with Luis Barrero here and the Department of Genetics in Groningen. And then, of course, a very, very special thank you to celiac disease patients and their families, without whom we would not be able to perform all these studies. Thank you very much.
Okay, thank you, Bonna. That was a wonderful introduction to celiac disease and the work that you've done to move us forward, particularly with the mouse model that develops atrophy, because that has been a problem for years, and you solved that puzzle. So hopefully more puzzles will be solved. My next speaker is Dr. Mark Anderson. He is a professor and director at UCSF Medical Scientist Training Program, part of the UCSF Diabetes Center, and he also is a University of Chicago alumni. So welcome, Dr. Anderson. His talk is Insights into the Pathogenesis of Autoimmune Disorders.
Thank you. Just takes a minute for my slides to kick up. Does it look okay?
Yes.
We're good? All right. Well, I just want to thank the organizers for inviting me to speak. And as it was mentioned, I am an alumnus of the UC and very fond of my training in Hyde Park. And so I am a physician scientist, so I see patients in adult diabetes and endocrinology with a focus on autoimmune disorders and why they happen. And so in my talk today, I thought I would give sort of a translational perspective on one aspect of autoimmunity and how it's really allowed us to have some key insights into how autoimmunity unravels. So before I go into the details of my talk, these are my disclosures. And you're going to hear more about this theme later today throughout, I think, many of the talks. So when we talk about autoimmune disorders at a high level, they are a complex set of diseases. And we think that the cause of autoimmune disorders is through a mix of both genetic and environmental factors. And to try to distill down things into perhaps a little bit more of a simplistic model about the genetics of autoimmunity, there is a lot of information to gather from identical twin studies. So in my area, in type 1 diabetes, we've followed enough identical twins that have developed type 1 diabetes to have an idea about the lifetime risk for the other identical twin, something we call the concordance rate. And as you can see here, we think that the concordance rate in type 1 diabetes for identical twins is around 50%. And likewise, similar data exists in rheumatoid arthritis. However, the penetrance in the second identical twin is not as high. But overall, people, when they see this data, would say, well, the other 50% is environment. And that may not be completely the case. And so one important thing to point out is the immune systems that are present in identical twins are not identical. There is a stochastic or somewhat random process in how the T cells and B cells generate their receptor repertoire that happens after a child is born and results in slightly different immune systems in identical twins. And so that other 50% could be related to this randomness. Well, despite saying all of that, we've had rapid advances over the last 20 years in our genetic understanding of autoimmune diseases, including celiac disease, by performing what's called genome-wide association studies. And this has allowed us to find many other genes that contribute to autoimmunity. But the overall theme is that this has been quite a challenge to unravel at a biological level. And that is because many of the genes that have been associated with autoimmunity have weak influence on disease. And you'll hear more about that later today. So perhaps a different approach to think about unraveling autoimmunity from a genetic perspective is to go and identify individuals that perhaps have a more extreme form of autoimmunity that may be related to a single gene. And so just want to present here a clinical case of a patient with a disorder that our lab has been working on for over 20 years. And this is a rare disease. In endocrinology, we refer to the disease as autoimmune polyglandular syndrome type 1 or APS1. And just to give you a flavor of the disease, I'm showing you a single patient case here that I had the opportunity of being involved in a number of years ago. So this is a 51-year-old woman who at the age of 7 developed calcium problems and was diagnosed with poor function of her parathyroid glands. At the age of 8, she started developing recurrent episodes of candida infections in her mouth and in her fingertips. And then at the age of 13, she lost the function of her adrenal glands and was diagnosed with an endocrine disorder called Addison's disease. At the age of 16, she lost the function of her ovaries and stopped having her periods and was diagnosed with ovarian failure. At the age of 20, she lost her hair follicles and developed alopecia areata universalis. And then finally, at the age of 40, she developed autoimmune type 1 diabetes. And all of the disorders that I'm listing on this slide here turn out to be autoimmune in nature, including the Candida. And the other striking feature of this particular disease, despite its rarity, was that it was appreciated by endocrinologists and other immune deficiency docs that this was being inherited in a simple Mendelian form. That was that the patient was inheriting a bad copy of one gene from their mother and their father. So an autosomal recessive disease. And so in the 90s, families that had children with this disorder were collected and genetic mapping was performed and the defective gene was identified using this technique called linkage. And so here rather than multiple genes summing to give you a risk for a disease like celiac disease or type 1 diabetes. Here, it's the absence of one single gene and you develop all of these autoimmune problems. And so to make a long story short, the gene was agreed to be called AIR, which stands for autoimmune regulator, that's defective in the syndrome. And so the big question, of course, is what does AIR do? And that's summarized here in this slide. And so we think we now think that AIR's major area of activity is in the thymus. And so the thymus is the organ that T cells get their name from. And so T cells leave the bone marrow and they go into the thymus, which is in the anterior part of our chest, and they go through an education process in the thymus. So these T cells commit to the lineage of being a T cell, and then each individual T cell rearranges a T cell receptor while they're in the thymus through this random stochastic process I referred to earlier. And this is a very powerful part of our immune system. This allows our body to have a diverse repertoire of different kinds of T cells that can see all kinds of things, including viruses, bacteria, fungi, and so on. But a downside to this stochastic process is that just by chance, some of the T cells that you're generating in the thymus are going to be reactive against yourself. And so what we think happens in the thymus is those self-reactive T cells are taught the difference between yourself and non-self. And this is where the AIR gene that's defective in this patient that I presented to you comes in. And so AIR is highly expressed in these special cells in the medulla of the thymus called medullary thymic epithelial cells. So they're not the T cells that are developing, but cells that the T cells that are developing are interacting with. And what AIR does inside these cells is it promotes the expression of what we call tissue restricted self-antigens, or TRAs. And what I mean by that is a gene or protein that's mainly only expressed in one particular location in your body. So a great example in my field in type 1 diabetes would be insulin. Insulin is only expressed in the islets of the pancreas and nowhere else in the body. Well, it turns out that insulin is actually expressed in the thymus. Not nearly as high as it is expressed in pancreatic beta cells, but it is made in the thymus. And we now know that AIR is promoting the expression of insulin. And along with insulin, many other genes that have this pattern, that is that they're made in one particular place in your body. So the way to think about it is eye proteins, brain proteins, liver proteins, heart proteins, and so on are actually made in your thymus and to some extent controlled by this very interesting gene regulator called AIR. And so this promotes the expression of these genes so they can be displayed to T cells while they're developing in the thymus. And then if autoreactive or self-reactive T cell encounters that while they're developing in the thymus, they get a signal to die and they're filtered out of your immune system. So now if you think about the patient that has the mutation in AIR, there's a failure to do this, and these self-reactive T cells can then escape out of the thymus and get into our mature immune system and create havoc, where they then go on to promote all kinds of autoimmune problems. And because AIR regulates thousands of these different kinds of proteins, you can see why a patient might develop different kinds of autoimmune problems. So with that background, I just wanted to touch on a few things that we've been doing related to this that have been very instructive about how immune tolerance works. And so as you saw in the slide, we had a patient that had multiple autoimmune features. And this is just a slide showing maybe at a deeper level, all the different kinds of autoimmune problems that can happen in these patients that have a defect in AIR. And so along with the autoimmune features that are listed in this slide, one thing that traffics with this is the patients develop not only immune infiltrates in these various organs that are shown here, but they also develop autoantibodies. And so these patients are actually a rich resource for identifying autoantibodies that we can use diagnostically, not only in these patients, but translate it to other diseases. And so one of the things that we've been doing recently that's very exciting at UCSF is I've developed a collaboration with my colleague, Joe DeRisi, who is the head of our Chan Zuckerberg BioHub effort at UCSF that some of you may be aware of. And together we have been jointly mentoring a UCSF MD PhD student, Sarah Vasquez. And the reason that we joined forces is Joe had developed an interesting platform to mine autoantibodies for their specificity. And without going into too much detail, why this is an exciting technique is it's sort of a new generation technique for autoantibody discovery. And so what we have generated in the lab is a phage library, which is a virus-based system that works in bacteria. So phage infect bacteria, and you can use them as a cargo, in a sense, to express different proteins. And so using a combination of bioinformatics, which is shown in the top left here, and molecular biology, we've been able to generate a virus library that encodes fragments of every single possible protein in the entire human gene, the entire coding human genome. And so each little virus is containing a fragment from a particular protein. The next fragment is from the same protein, but we've shifted over a little bit. And so in our library, we have over 700,000 unique phage that have these little fragments. And the reason that this is very powerful then is it's an unbiased library that we can then use the serum from the patients to screen and identify potential specificities. So another key part of this technique that also has allowed this to emerge is something that we're using more frequently in the laboratory is next generation sequencing. And so we have access to very cheap next generation sequencing that allows us to sequence millions of base pairs of DNA at a relatively low cost. So the way this technique works is you can take antibodies from either a different source or a patient, you incubate it with the library, and the antibodies interact with these viral phage particles that are expressing little fragments of the entire human coding genome. And then you pull down the phage with the antibodies, purify them, and then we use sequencing to identify what is the polypeptide sequence that these antibodies are binding to. And so together, this has turned out to be a great technique to identify new autoantigens in this clinical disorder. And so what I'm showing you here in this very busy slide is just a little heat map of what we pulled out of screening at this time, approximately 30 or 40 AIR mutant patients or APS1 patients compared to healthy control. And then each row in this heat map on the top are proteins that we knew AIR patients made autoantibodies to. And so if you just look at it at a 10,000 foot level, you can see it works very nicely overall in that we find many of the things we would expect to find in APS1 patients that are completely absent in healthy controls. And then there's some things on this list that actually make a lot of sense. So there's this gene here, CYP11A1, you can see is present in many patients. And that is a gene that's highly expressed in the adrenal glands. And many of these patients get adrenal autoimmunity and so on. Okay, so this is the slide that's actually the more interesting part, of course, is the discovery slide. And so what I'm showing you here is the same kind of heat map. We're identifying autoantigens through this technique that appear to be robustly enriched in the APS1 patients compared to normal controls. And so each row is a unique gene. And then all of the dark genes in these rows are new things that had never been identified before in these patients. And so using this technique, we think that we've identified over 50 different autoantigens that are present in APS1 patients. And of course, I don't have time to go through all 50, but it underscores the power of this technique in identifying these autoantigens. And so I just wanted to tell you a little bit about a couple of the autoantigens that we pulled out in this screen. So here, what I'm showing you is taking all of the APS1 patients together and correlating whether they had an autoantibody with a particular clinical phenotype or clinical problem that these patients had. And so one of the ones to look at here is this new autoantibody we identified against a protein called KHDC3L. And so what it correlates with in this correlation plot that we've made is POI. And POI is primary ovarian insufficiency. And it turns out that KHDC3L is an ovarian specific protein. And then we've gone on to validate this in our cohort where we've segregated patients into whether or not they had ovarian insufficiency, so negative versus positive. And you can see that in the positives, we have enrichment for the presence of these autoantibodies. Okay, so a couple of other ones that I wanted to highlight real quickly. So an unusual phenotype in these patients that no one's really completely understood is many of them get problems with enamel hypoplasia in their teeth. They lose the function of their enamel. And nobody really understood why this was. And so one of the hits that we got in this screen were autoantibodies against this protein called ACPT. And in some ways, ACPT is a little bit of a misnomer with the word testicular in its name. It is expressed in the testes, but the place where it is most highly expressed is actually in the developing teeth. And we think that this is actually why patients develop enamel hypoplasia in this disorder, because human beings that are knocked out for this particular enzyme also develop enamel hypoplasia. And then coming around to today's session related to celiac disease, one of the features in air deficient patients or APS1 patients is many of them develop intestinal dysfunction. And when they have and they don't develop celiac disease, but when they have intestinal biopsies done, when they have intestinal dysfunction that includes abdominal pain, malabsorption, chronic diarrhea, one of the things that's been noticed by many clinicians with these patients is a particular kind of cell in the intestinal epithelium can be missing in these patients. And these cells we call enteroendocrine cells. And these are specialized cells in our intestines that make endocrine hormones that then communicate with other organs. And so examples of this include things like cholecystokinin, which is a hormone that communicates between the small intestine and our gallbladder and tells the gallbladder to start contracting. So a really unusual thing about these AIR mutant patients is often these cells are missing. And we actually think that on our screen, we found why this is. And so in many of the AIR mutant patients, they develop autoantibodies to a protein called RFX6. And so if you look at perhaps the bottom here, just cutting to the long and short of it, we segregated our AIR mutant patients into whether or not they did not have intestinal dysfunction versus they did have intestinal dysfunction, and then whether or not they have autoantibodies to RFX6. And you can see a great enrichment for the presence of autoantibodies to RFX6 in APS1 patients. And so what about the RFX6 protein? Well, it turns out that RFX6 is actually an important factor for the development of these specialized cells in the intestine. So if you go back up here to the top, we're now staining a section of small intestinal tissue with control serum, an APS1 patient that does not have these antibodies, and then an APS1 patient that does have these antibodies. The key thing that you're looking for is the green and red coming together that gives you a yellow color. And so you can see these specialized cells right here in this box that are then highlighted at higher power are being targeted by autoantibodies in these APS1 patients. And we think that this leads to the loss of the function of these cells. And so the overall arc that I'm trying to give you here is that this is a very powerful technique to go and mine autoantibodies, and that we found some new targets. And we actually think that we found a target that explains intestinal dysfunction in these patients. And similar, I think, since this audience is interested in celiac disease, I think a very important thing that has developed in celiac disease, of course, is there are autoantibodies to transglutaminase and other specificities that are very helpful in helping make the diagnosis of celiac disease. And we now think that this RFX6 autoantibody could be used in patients that come to clinic that have intestinal dysfunction that may not be caused by celiac disease. Another important highlight I would make about the technique is that it's relatively inexpensive. And so in our hands in the laboratory to run a sample through this screening process at a high throughput level, we now think costs less than $5. And so we're very interested in collaborative efforts to look into other autoimmune diseases and other autoimmune patients. So something that I wanted to touch on, given the times that we live in right now, is a point related to COVID. That was completely unexpected in this story that's rapidly emerged actually in the last couple of months that I think you'll like to hear about. And so this is a slide I already showed you, but one of the features of AIR defective patients is they happen to make autoantibodies to particular kinds of cytokines called type 1 interferons. And so that's depicted in this slide here. So if you look at this busy slide where it says interferon A6, A2, and A5, these are type 1 interferons. And so these are signaling cytokines that are very important in antiviral responses. And so one of the things that's emerged in the pandemic that's not widely published but has been appreciated is patients with this disorder that happen to have gotten infected with SARS-CoV-2 have had a poor clinical course with COVID-19. And so this led to investigating whether or not these autoantibodies were present more widely in patients that are coming to the hospital with COVID-19. And this was just recently published in Science in a collaborative effort that was led by Jean-Laurent Casanova at the Rockefeller. And we feel very fortunate to collaborate with Jean-Laurent. But if you look at this slide here, in the top left, now each row is a patient. Except for this row here, APS1 patients, which are a positive control for having these autoantibodies to type 1 interferons. And what Jean-Laurent Casanova's group had figured out is roughly 10 to 14% of patients in the intensive care unit who are severely ill with COVID harbor these autoantibodies. And it's likely that these autoantibodies are inhibiting the patient's ability to respond to COVID-19. And so we have not published this data yet, but we've looked in patients ill with COVID in San Francisco. And the Casanova lab was kind enough to share their samples with us in San Francisco just to validate our assay against interferon alpha A2, one of the type 1 interferons. And then if you look over on the right here, we're now exploring the frequency of these autoantibodies in subjects in San Francisco that have developed or that have had COVID-19. And you can see that we do pluck out a subset of patients that are positive for these autoantibodies in these two cohorts from San Francisco. But if you go down here into the bottom, this is where things get more interesting. So overall, we find, roughly 3 to 8%, so around 5% of all comers are positive for these autoantibodies, but they're greatly enriched in patients in the ICU. So we find that somewhere around 13 to 19% of patients in the ICU in San Francisco have these autoantibodies, and it does correlate with a poor clinical course. And so this is a rapidly emerging area, and it makes sense that these autoantibodies are inhibiting the ability to respond to the virus because these interferons are important in the viral response. But the question is, of course, where did they come from? And we're not completely clear on that right now. One clue is that most of the patients that develop these autoantibodies are men. And the second thing is that we've gone and screened in the general population, and we found that around 0.3% of people in the general population harbor these autoantibodies. And so this has led to some a new biomarker that we could look at that connects autoimmunity to COVID-19, and perhaps somewhat of a surprise that there is a connection there. So with that, I just have one other quick slide I wanted to show, and then I'll wrap up. And that was going back to the thymus. And remember that I told you that air is highly expressed in the thymus to educate our T cells against self. And so these cells that exist in the thymus have been difficult to study in human beings, but we've had a rapid emergence of a powerful technique called single cell RNA sequencing that we have utilized on the human thymus. And so this is just a little diagram of how this all works. But the reason this is important is we can now characterize cells which are much higher resolution than we'd ever able to do before. And just to jump to the chase, not only have been able to do this, but we now have been able to identify the air expressing cells in the human thymus and what they're doing. And so if we jump here, this is my last data slide. What I want to show you here is each little dot in this this fancy plot represents a cell, an epithelial cell from a human thymus, and the air expressing cells are located right here. Okay, and then if we take all those autoantigens that I told you about earlier that we identified in the paper as a group, gave them a score, where are they expressed in the thymus? Well, lo and behold, they do light up specifically in this population. And then to just bring it back home to where I started at the beginning of all of this, if we look for the actual expression of insulin in the human thymus, it is there. And it turns out that it's expressed specifically in the cells that make air. And so with that, I'll just thank all the people that contributed to this work that I told you about today. We're going to particularly highlight Sarah, who had done the beautiful autoantibody work, and Joe for his willingness to collaborate on it. I'd also like to thank John Laurent Casanova and Paul Bestard from his group that have done this really interesting work on the type 1 interferons. Thank you.
Okay. Thank you, Mark, for that talk. You got me thinking about malabsorption and some of the mysterious patients I see where we can't explain it. So I'm going to think about air. Our next speaker is Gary Firestein. He is a distinguished professor of medicine at the University of San Diego in California. And he is the Dean and Associate Vice Chancellor for Clinical and Translational Research. And his talk is entitled Rheumatoid Arthritis, the Intersection of Genetics and Epigenetics. Welcome, Dr. Firestein.
Thank you very much, and I appreciate the opportunity to talk a little bit about our data. These are already hard acts to follow, fantastic talks and great science. We're going to shift gears a little bit and talk about rheumatoid arthritis, which is an immune-mediated disease that occurs in about 1% of our population. Here are my disclosures. So RA, as I mentioned, occurs in about 1% of the population. It's more common in women than men, sort of usually a 2.5 or 3-to-1 ratio. And it's a symmetric polyarticular arthritis, which means it involves particularly the small joints of the hands and feet, typically many joints. Shown here is the classic presentation of metacarpal phalangeal joints and proximal interphalangeal joints, but ultimately can affect wrists, knees, hips, and so on. It's actually there's been a huge amount of progress over the course of the last couple of decades, not only in terms of understanding pathogenesis, but also in terms of management. And I was hearing and learning actually about altered peptides and antigens in celiac disease, and an analogous system occurs in rheumatoid arthritis, where at least the beginning of the disease is not related necessarily to true autoantibodies or autoantigens, but to modified peptides. And what happens in the earliest stages, even in pre-RA, is that the peptidyl arginine deaminases can modify peptides by converting arginine to citrulline. And those antibodies that develop to those altered peptides appear oftentimes 5 or 10 years before the onset of disease. And those antibodies are not in and of themselves pathogenic. They don't induce arthritis, but they're clearly a marker for a disease that can subsequently occur. The other thing that I just wanted to briefly comment on is the heterogeneity of the mechanisms of disease for RA. We have highly targeted agents that have efficacy in this disease: TNF blockers, B-cell depleters, T-cell co-stimulation inhibitors, and they are in fact effective, but each of them is effective only in a subset of patients. About a third of people will get dramatically better, a third somewhat better, and a third not better at all. And each targeted therapy will have benefit in a different subgroup of patients. So in some ways, I think it's better to think of RA as a syndrome with a common clinical manifestation that has many pathways. And I'm going to come back to how we're trying to understand that diversity of mechanisms in a few minutes. Well, we already heard a little bit about disease concordance in twins, and it is concordant in rheumatoid arthritis. The number I generally use is 12 to 15% concordance in identical twins, and it is quite correct that there is a certain degree of randomness in the development of the immune system, and there are other influences such as epigenetics besides the environment that may differ between identical twins. But compared to other diseases like type 1 diabetes or lupus, this is really significantly lower. So at least 80% of susceptibility and severity is due to non-coding influence. That's kind of how I like to view it. And some of it could be environment, and the best described of those is smoking. It clearly, as if we needed another reason not to smoke, it is a clear environmental risk factor for RA. There's an emerging literature on the microbiome as well. But this notion that non-coding influences are important led us to study epigenetics, which are heritable, but also plastic changes in gene activity that are not caused by changes in DNA sequences. And this oftentimes can integrate non-genetic influences and the environment with regard to risk. And there's a large number of epigenetic mechanisms. The ones that most people talk about and study are things like DNA methylation, where there are covalent bonds that alter can alter DNA and gene regulation. Histone modifications and microRNA are some examples. So our story with regard to epigenetics and RA really began by studying DNA methylation in patients with rheumatoid arthritis. And our favorite cell to study are synovial fibroblasts, sometimes called fibroblast-like synoviocytes. Sometimes we just call them synoviocytes. And several years ago, we began exploring DNA methylation patterns in synoviocytes isolated from the joints of patients with rheumatoid arthritis and other diseases to try to understand if there were certain patterns. And this shows one of the larger data sets that we've published, although we're actually now up to well over 50 or 60 lines that we've studied in detail. We use as a control osteoarthritis for DNA methylation, and for most of our synoviocyte and synovial tissue work, as you can probably imagine, it's not particularly easy to get synovium from someone who is normal pre-autopsy. So the OA is a common surrogate. And the observation here, if you look at this heat map, each of the horizontal lines is a locus for DNA methylation. Osteoarthritis, which is non-inflammatory in general, or at least not immune-mediated in general, segregate together, except for a couple of minor exceptions. And rheumatoid arthritis forms its own separate group. And so we have published actually quite extensively on this, that these differences in DNA methylation are related to many pathways that we're all familiar with related to immunity, including cell recruitment, cell adhesion, cell growth and differentiation, and so on. This is not a fixed change. In fact, we've looked at very early rheumatoid arthritis, people that have less than a few months of disease. They are also different from osteoarthritis, but they also form a separate subgroup compared to rheumatoid arthritis. So what this means is that there are clearly differences between RA and non-RA, and the system evolves over time. You begin to accumulate marks that distinguish RA, and we think that this is something that is very important in terms of how the disease changes as it progresses, as it becomes increasingly aggressive and increasingly destructive when not treated, or when inadequately treated is really, to be more precise. So, so the important lesson here is differences in epigenetics between RA and non-RA, and that it is a process that evolves over time. After these initial observations, we moved into much larger data sets and have done whole genome unbiased approaches where we gathered information on the transcriptome, DNA methylation, and instead of using DNA chips, we did whole genome bisulfite sequencing, a panel of histone marks through using a technique called ChIP-seq, and these marks can open up chromatin and allow genes to be expressed, or they can repress expression depending on which particular marks and how they're altered. and then open chromatin through a technique called ataxia, which measures typically areas that are either ready to or already being rapidly expressed. And I'm going to cut to the chase on this one. There's a huge amount of data that has been published, actually about 12 terabytes of data from 22 different cell lines. And the bottom line is really exemplified in this particular slide. When you look at what my colleagues and I developed a novel algorithm called EpiSig that can integrate all of these different epigenetic marks. And so you can identify individual differentially epigenetically modified regions. And the part that is abnormal in rheumatoid arthritis is gene transcription. It is a highly activated state. Now, I know that that may sound obvious in retrospect, but what we weren't clear was whether RA and its high transcriptional activity was due to overactive promoters and enhancers, or whether it was because repressive mechanisms were inadequate. And it turns out it's the former. And so these marks here, H3K4 monomethyl and H3K27 acetylated marks, are active transcription marks for promoters and enhancers. And they are quite elevated in rheumatoid arthritis. And these are repressive marks here, and they are just a rounding error in the epigenome in rheumatoid arthritis. Open chromatin, which really is, I think, a reflection more of these particular marks here, is also increased. Now, we found a number of expected and unexpected pathways that are differentially modified in RA. Some of them we expected. We'd already done quite a bit of work in p53 signaling, integrin signaling, and other immune responses, and those are differentially marked in rheumatoid arthritis. We also found some unexpected pathways, and the one that got the most attention when this was initially published was Huntington's disease. And so there are shared epigenetic marks between Huntington's disease and rheumatoid arthritis and shared pathways. And so this creates many opportunities for trying to understand commonalities between diseases that clearly have different phenotypes and we thought had very, very different mechanisms. But it does enable us to think in broader terms about the taxonomy of diseases based less on the clinical phenotype and more on fundamental cellular mechanisms. More recently, we've been looking at the peripheral blood to see how that also reflects upon abnormalities. Of course, the synovium in rheumatoid arthritis is shown here on the right. This is the lining of the joint. This is the inside of the joint. It is a highly immunologically active tissue. And in peripheral blood, there are cells that are trafficking back and forth between RA synovium. And so in order to try to understand whether we can use the blood as a mirror to look at the joints, we've now collected and studied a series of matched T cells, in this case, from patients with RA at the time of joint surgery and determined their epigenetic patterns, and in this case, DNA methylation. And I think here we go. So this is a principal component analysis. And what it shows down here are the peripheral blood cells from patients with RA in the red and OA, osteoarthritis, which is our control. And they actually kind of cluster together here, which is interesting. But if you look in synovium, they are actually quite different from the T cells that are in the peripheral blood. And we were somewhat surprised by the extent of the differences between them. So the principal difference and the principal influence on DNA methylation in rheumatoid arthritis and in fact in osteoarthritis is where the cells have accumulated. And when we look at those differences in more detail, the differences between RA blood and RA synovium are highly enriched for immune processes. But if we look at osteoarthritis, we see actually, although there's plenty of genes, they're fairly random in terms of their distribution. So what this means is that location, like real estate, this is also very important for DNA methylation and epigenetic marks, and the location is the most important determinant. And there are methylation abnormalities that are readily identified in both RA and OA, but the ones that are in RA are specifically selected and enriched for processes involved with immune responses and inflammation. In contrast, in osteoarthritis, it is random. And this is actually, I don't have time to go into that in great detail, but this is a recurring pattern, is that OA has a lot of abnormalities. They're basically random. In rheumatoid arthritis, they are clearly directed towards immunologic diseases. So where do we go from here? And for this, I want to talk a little bit about, give a brief background on applying lessons from Google and Spotify and how we are beginning to integrate this and determine how individual patients are different from one another. So Google uses a method called PageRank to rank websites. And I thought it had something to do with web pages originally. It turns out Larry Page was the founder of Google, and it's called PageRank because it's named after Larry Page. And if you do a Google search, it actually goes through and searches the internet for different websites and how they interact with each other. And then it ranks them based on how many times other websites refer to them, the quality of those interactions, and other influences. So for example, if you did a website for different colored happy faces, you can see there's lots of different flavors here. The big yellow guy here has multiple connections all over the place, and it is a big yellow ball here. And so in a Google search, this would be number one, be the top hit. The blue guy is also very important. It has lots of interactions, not quite as much as yellow. It's next, followed by red, followed by green. So that's actually how when you're doing a Google search, that's how it ranks these things. And of course, you can game this by clicking multiple times on some of these other websites, but this is in general how the internet and Google will decide which are the most important things for you to look at. Well, we can actually do the same thing with gene networks. And this is called the Tai Chi pipeline, developed by my colleague and collaborator, Wei Wang. And it is a combination of RNA-seq and ATAC-seq. So remember, ATAC-seq is open chromatin. And what we do initially is we do ATAC-seq, and these are studies on cultured fibroblast-like synoviocytes from the joints of patients with rheumatoid arthritis. And we look at all of the areas of open chromatin, and we find which ones have transcription factor binding motifs. Transcription factors are the proteins that play a critical role in initiating the complexes that lead to gene transcription. We then look, so in this case, here are examples of transcription factors. And then we look at what are the genes that should be regulated by those transcription factors. And we develop this network. So some transcription factors regulate other transcription factors, some regulate different genes. So we then, instead of trying to predict which genes are expressed, we use RNA-seq to look at the actual transcriptome. And we look at which genes are actually expressed the most. So here, this particular gene is expressed a lot. This one a lot. Some of these others, not quite so much. And so now we have this network that is somewhat weighted based on the actual expression of genes. And then the final step, the PageRank, just like a Google search, is we look at which of these transcription factors have the most connections to the most highly expressed genes. And if you do that, you could then rank these. And this one would be the most important because it has the most connections to the most highly expressed genes. And these are next and followed by this guy. So we can then make a rank. So before I get into the data, the next step is actually similar to Spotify. So anyone from my generation doesn't have a clue what Spotify is, but I have learned a lot more about it recently, and it's really a way of collecting what are called playlists of songs. And so, for example, if you have two people, David and Kathy, for example, David has this list of songs that he likes. Kathy has this list of songs, and they overlap, but they have some differences. And this shows how they overlap. Now, what Spotify does is it says, well, if Kathy likes G and they have so many similarities, David probably will like G as well. And the same thing for A, song A. And so what it does is then create a master playlist for David and Kathy that allows them to expand their repertoire of songs that they might like. And over here is a third person who has very different likes, and that's an entirely separate playlist. We could do that with transcription factors as well. For example, David has this set of transcription factors, and I've kappa B, AP, and so on. Kathy has this set. They have overlap, and we can create a transcription factor playlist for them that includes not only the areas where that they have in common, but also areas where they don't necessarily have their own specific. We can't detect a preference for a transcription factor. When we do this, we were very surprised to find that transcription factors and the transcriptome can separate RA patients, or at least their synoviocytes, into two large clusters, shown here cleverly named by us cluster 1 and cluster 2 with this heat map. And the next step that we did, that we took at this point was to try to understand how these clusters are different. And what we did was we used the informatics tools to try to predict different transcription factors that would have the opposite function in cluster 1 versus cluster 2. Now, this is previously completely undescribed, this degree of context-specific transcription factor function. But we were able to find actually quite a few of these where a transcription factor would be predicted to be stimulatory in cluster 1 patients and suppressive in cluster 2 patients. And the one that was the top of the list was retinoic acid receptor alpha, which is involved in cell differentiation and proliferation and a variety of other factors. And this shows of the cell lines that we looked at from cluster 1 and cluster 2 cells. They're supposed to have the exact opposite effect. So this is a hypothesis that we could test because RARA regulates TGF beta. And so we can knock down RARA in cluster 1 and cluster 2 cells. And much to our, or at least my complete surprise, it actually worked. If you knock down RARA in cluster 1 synoviocytes, the TGF beta expression goes in one direction. If you do it in the other set of synoviocytes, the cluster 2 cells, it goes the opposite direction. And it's not just that TGF beta goes in opposite directions. You could march up and down the entire RARA TGF beta signaling pathway, including SMADs, CDKs, all the way down to RB1. And you can find that the entire biology of RARA is different in cluster 1 cells than in cluster 2 cells. So what does this mean? Well, I mentioned to you that there are very different mechanisms of disease in different patients with RA. And that what we now know is that genes and networks can now vary between these patients. Transcription factors can have very different functions in individual patients. RARA is just one of them. We have a long list of these that we're busy testing right now. But we can use this now to potentially begin to stratify patients into different disease mechanisms, either to target specific transcription factors or their pathways, or in order to stratify people into which of the existing therapeutics might potentially be amenable to. So this could provide clues to the elusive Holy Grail, a rational approach to personalized therapy. because right now it is really it is a random, to our eye at least, a random walk through the clinic where we have an algorithm where we sequentially try and use trial and error with different targeted approaches. So that is an area where we are actively working right now. Take-home messages: there's more to disease risk than genetics, or at least for RA. The environment matters, and I didn't get a chance to talk much about Lamarck versus Darwinism, but Lamarck was actually partly correct. Location matters, as I mentioned before. Patients differ from each other. Gene networks have a lot in common with Google and Spotify. I didn't really talk a lot about RA and when it actually begins, but a lot of this stuff actually occurs before people have symptoms. They have what we call pre-RA, similar to pre-diabetes. And we are currently mapping the epigenome in those individuals as well. This was the work of a large number of people, and I list some of them here. Wei Wang, I already mentioned. David Boyle, Professor Boyle has worked with me and collaborated for many years. We have some wonderful sponsors who have supported us. And in Colorado, for the pre-RA project, we are collaborating with Colorado group, Kevin Dean particularly, and Mike Hollers. And with that, I thank you for your attention. And I think I finished almost exactly on time.
Almost, not quite, but you're pretty good. Thank you for that talk. Again, outstanding talk so far in my session. I'm thrilled. My next speaker is Dr. Luis Barrero. I'm sorry. He is an associate professor of medicine. He's on the Committee on Genetics, Genomics, and System Biology here at the University of Chicago. And he will speak to positive selection of genes in autoimmune disorders.
Hello, good morning, everyone. Thanks so much for the invitation. So today, my talk is going to be a little different from what you have seen so far this morning. It's not going to be focused on any particular disease, but more broadly on, trying to think a little bit more about, how past evolutionary events might have contributed to present-day variation in susceptibility to infectious diseases and autoimmune disorders. So if I have no conflicts to declare. So I'd like to start by making the point that, as an evolutionary biologist, you tend to think about what are the different forces that might contribute to the evolution of the human genome. And one of the things that we believe to have been a major contributor is the selective pressure imposed by pathogens. And that, I think, it's well translated by just looking at historical survival curves. So that's what I'm showing you here in this plot. So on the x-axis, you have age. On the y-axis, it's survival rates. And I'm showing you two curves. So survival rates in the Neolithic and up to the 19th century in the UK. And as you can tell, these curves have hardly changed over most of our evolutionary history. And by the age of 25, 30 years old, 50% of the individuals will die. Then in a very short evolutionary period, we started being able to control and manage the pathogens in a better way. And that was driven by many events, but probably some of the most important were some of the discoveries from Louis Pasteur, the discovery of the rabies vaccine that was followed by discovery of many other vaccines, and then more recently, the discovery of antibiotics. So in a very short period of time, we started being able to control pathogens in a way that was not possible before. And you go from survival curves that look like this to something that is going to look like this. So I think this is pretty strong evidence that pathogens have imposed a very strong selective pressure in the human genome. So one way that you can approach and try to identify the contribution of natural selection to the evolution of the human genome is by looking at patterns of genetic diversity. And the reason why that works is that I'm going to kind of go through this in a very schematic way. So imagine that in the population, you have a certain level of genetic diversity that is illustrated here by these different brown dots. So imagine these are polymorphic variants in the population. Each line, it's an individual. And now imagine that there's a mutation, a new mutation that appears here in red that confers you protection against a certain infectious disease. So what will happen is that individuals that have this mutation are more protective, so they are better able to survive. And as a result, what will happen is that over evolutionary time, this mutation and the mutations that are associated with it will rapidly increase in allele frequency up to the point that everyone in the population now has this mutation, right? So the point is that the patterns of genetic diversity, if you compare them prior to the selective event, so under neutrality, and after positive selection, they are going to look, I think you will agree, very, very different. And these differences in patterns of genetic diversity can be identified through what we call neutrality tests. And that's how, that's one of the approaches that we can use to identify the contribution of natural selection to our recent evolutionary history. So when you apply these neutrality tests genome-wide, one thing that comes across from this analysis that both at the interspecies level, so when you can try to identify selective events across different species, or within human populations, basically the biological terms and the genes that are the most enriched for these signatures of positive selection are things that are associated with immune responses, which again strongly supports this notion that pathogens have been a really important selective pressure throughout our evolutionary history. So among the many events that have contributed, that potentially have contributed to the evolution of the immune system, one that arguably was very important was the out of Africa exodus. So for, probably most of you already know this, but we are actually a very recent species. So Homo sapiens appeared in Africa only about 200,000 years ago. And then more recently, about 60,000 years ago, there's a group of individuals that left Africa to colonize the rest of the globe. So all the individuals that live today outside Africa are really the descendants of this group of individuals that left Africa only 50,000 years ago. So as a result of these migratory events, we can speculate that humans have been exposed and had to adapt to new pathogenic realities. And if that is true, one could also speculate that as a result of those adaptive events, the immune systems of Europeans versus African ancestry individuals, for example, might have diverged. So in the lab, we are very interested in formally testing those hypotheses using a combination of functional genomic studies and population genetics. And so one of the first steps that we performed was as following. So it's a relatively straightforward experimental design. So like I said, we are not really interested in any particular disease. So we are really interested in understanding variation in immune processes in the general population, as the individuals that live today are really the result of those that survive all these selective pressures through our evolutionary history. So we collected blood from a panel of 175 individuals that varied in their level of European and African ancestry. From these individuals, from these blood samples, we derived macrophages that are important in immune cells to fight pathogens. And then in vitro, we infected these macrophages with two live bacteria, so in this case, Listeria and Salmonella. So the readout of this experiment is RNA sequencing, which has already mentioned before, that allows us to measure gene expression levels across the entire genome in non-infected and infected cells. And then we also had genotyping information from each of the individuals that allows us to correlate particular genetic variants with variation in immune response. So with the genetic information, we can identify what's the proportion of the gene of each of these individuals that is of African or European ancestry. So that's what this plot is trying to show here. This is a structure plot. So these are individuals that self-identify as being African Americans, and as you can tell, so each column it has two colors. So the color green represents what's the proportion of the genome of the individual that is of African ancestry, and the pink of European ancestry. So for example, an individual in this area, it's an individual that will have 75% of the genome of African ancestry and 25% of European ancestry. So as expected, for example, we see that self-identified African-American individuals have in average about 26% of European ancestry in their genome. So now that we have this continuous variable of proportion of African ancestry or European ancestry, if you will, it's the same thing, we can start asking a simple question. Is there any relationship between genetic ancestry here in the x-axis and, for example, the magnitude of the response that you engage in response to these two bacteria. And this is what we call here, for example, population differently responsive genes. So I'm not going to go into the details of this test, but what we have shown was that over 20% of the genes that respond to either infection show a significant difference in the intensity of the response between European and African ancestry individuals. And I think something that was quite surprising at the time was to see that there was this very clear trend whereby increased African ancestry was associated with a much stronger transcriptional response to infection. We went further and we are actually able to identify what was the exact genetic basis for some of these differences. And in average, we estimate that 30% of the population differences in immune regulation are explained by change in allele frequencies of expression quantitative trait loci, which I'm going to explain a little bit more in detail in a second. But basically the point being that we believe that there's really an important genetic contribution to some of these differences. So this was interesting, but now one of the limitations, of course, of this study is that we focused on a single cell type. So in this case, was macrophages, and therefore, you know, an immune response is really not just one single cell type working in isolation. It's really the result of the interaction of many different immune cell types, and also the crosstalk between innate and the adaptive immune system. So now with the advent of new technologies such as single cell RNA sequencing that Mark already mentioned earlier, we can start, you know, expanding this type of studies to really try to understand how genetic ancestry impacts the immune response in a cell type specific fashion without actually having to sort the cells and infect them in isolation. So this is a project that it's completely unpublished. So this is work from a PhD student here at the University of Chicago, Haley Randolph. So what she did is that she, again, very similar study design, she collected blood samples from a panel of 90 individuals in this particular case, also European and African ancestry individuals. But this time around, instead of isolating the particular cell type, we infected all peripheral blood mononuclear cells, so all the white blood cells that are in circulation. We infect them with flu, the pandemic strain of flu. And now what we are doing is that we are really allowing the different cell types to respond to this infection in a coordinated way. And then after the infection, we perform single cell RNA sequencing on the infected and non-infected cells. And by doing so now, in this particular case, we profiled about, you know, over 230,000 cells. So we have transcriptome, genome-wide transcriptome levels for all these, for over 200,000 cells in both mock and flu-infected conditions. And now, because you know, we have expression levels across all these genes, we can actually classify the different cell types that compose this PBMC population. And in this particular case, what we see is that, as expected, we find, you know, a population of CD4 T cells, CD8 T cells, natural killer cells, B cells, monocytes, and then smaller populations of NKT cells, dendritic cells, and some granulocytes. So now the interesting part of this is that though we have not been, we did not, you know, physically separated all these different cell types to study how individual they respond to this infection, but computationally we can identify what are the different cell types and specifically now ask the question, how did monocytes respond to the infection? How did B cells respond to the infection, and so forth? So when you do that, what we see is something that we'll probably have predicted in advance, is that there are, you know, many genes, so thousands of genes, so about, you know, over 20% of the entire genome. So here we measure gene expression levels in each of the cell populations for about 10,000 genes. And what we see is that all the different cell types are inducing a strong response to flu, with the cell type showing the strongest response being monocytes. And the reason why monocytes appear to respond the strongest is that these are probably the cells that are really getting the most infected with flu. And we can tell that because we can actually measure and quantify transcripts from the flu genome. And therefore, we have a very good proxy of viral load in each of the cells. And we can see very clearly that monocytes are the ones showing the most amounts of viral transcripts. So these are the ones that probably are going are getting the most infected. So most likely what's happening is that these are the cells that get the most infected, and then they are sending paracrine signals to the other cell types that is engaged, that is inducing the response that you are seeing here. So now using this data, we can start asking within each of these cell populations, how, if there's a relationship between genetic ancestry and the response to flu. So this is very similar to the slide I just showed you earlier. So we're just really looking at the correlation between genetic ancestry and gene expression level, or genetic ancestry and the magnitude of response that you see to flu. And what we see again is that there are actually thousands of genes again within each of these cell types that respond that correlate with genetic ancestry. So genetic ancestry, the effect sizes are relatively small, but it's quite ubiquitous. So genetic ancestry seems to play a quite significant role in variation in gene expression levels. Interestingly, if you ask whether these ancestry effects tend to be shared or cell type specific, what we found is that for the vast majority, they tend to be cell type specific. So over 50% of the ancestry effect that you observe are observed either in a 1 or 2 cell types at the most. So this is important to keep in mind, because it seems to suggest that characterizing the ancestry effects on a single cell type is really going to miss a lot of the true ancestry impact on the regulation of the immune system. So then we ask, what type of pathways and biological terms appear to be the most divergent between European and ancestry African individuals. And this is a quite busy figure, so I don't expect you to be able to understand it right away, but let's just focus on one particular pattern, that is the interferon response, which appears to be the most divergent between these two ancestry groups. So what we are seeing is that, for example, in monocytes before infection, so the red means that these genes tended to be more highly expressed in African ancestry individuals, and blue that the genes tend to be more highly expressed in European ancestry individuals. So what we are seeing is this very interesting pattern that at baseline, it seems that African ancestry individuals, they tend a higher baseline level of interferon responsive genes, but after infection, so once you infect them with flu, this pattern completely reverts, and now consistently across all cell types, you see that European ancestry individuals engage a stronger response to flu. So we kind of decided to follow up this more closely, as interferons are very important, and they actually they were mentioned in both Banner's talk and Mark's talk this morning as being incredibly important, as protective against response to virus, but also potentially pathogenic if response is uncontrolled. So we developed for each individual what I call here an interference score, that basically it's a score that tells you like how much what's the activity of the interferon pathways for each of these individuals, and see if that interference score correlates with genetic ancestry. And what we saw is, again, at baseline, there's very weak correlation, but after stimulating the cells with flu, we see this very striking relationship between genetic ancestry and this interference score, whereby the activity of interferon response is elevated in European ancestry individuals as compared to African ancestry individuals. Interestingly, this interference score is also very strongly correlated with viral load. So the more you engage of interferon, the stronger it's your viral load as well. And this, of course, ends up translating to the fact that individuals of European ancestry, which are the ones that engage the stronger interferon response, they are also the ones showing increased amount of viral transcripts. So then we wanted to understand what, if we could characterize the genetic basis of these differences, and to do so, since we are looking at differences in gene expression, the obvious thing to do is to try to identify what we call cis expression quantitative trait loci, or CISQTL. So this is kind of a schematic of what does that mean. So if you imagine that there's this particular gene here, HLA-C, for example, imagine that there's a polymorphism, a mutation in the promoter of this gene. So you can either have the T allele in your genome, or you can have the G allele. And now imagine that the T allele allows the binding of the transcription factor that is important to activate the regulation of this gene. So individuals that have the T allele will have a lot of gene expression, where the G allele disrupts that binding site and reduces expression. So in that situation, what you expect is that if you have the T mutation in your genome, you will have a lot of HLA-C. If you have the G, you will have very little of HLA-C. So you can do this genome-wide. So like I said, we have about 10,000 genes in each of the cell types, and we have two conditions, infected versus uninfected. So you can do this basically in all these different conditions. What the end result of this is that we identify what I call here E-genes. And E-genes are simply genes for which there's at least one genetic variant that seems to significantly impact their expression level. And across all these different conditions, we identified over 2,000 genes that appear to be to some extent genetically regulated. This is just a kind of an example of OAS1, which is a very important interferon inducible gene. So you see that for this particular gene, there's no EQTL. So this gene, there's no the genetic variation does not seem to impact the expression of the gene before infection, but it does impact the expression of the gene after infection. We see, as expected, suggesting that there's a strong genetic component to the ancestry effects that we see between populations, is that genes that are associated with genetic ancestry, they are also very strongly enriched for E genes, up to 6-fold different. So next we wanted to, am I running out of time?
Yes, you've got 2 minutes. Yeah, not even.
Okay.
Sorry.
So but I'm going to skip just to the last part. So we wanted to then understand whether natural selection. So now we basically are in a situation where we have a large number of genetic variants that appear to explain population differences in gene expression and immune regulation. And in the past, we had seen that we wanted to understand whether these might explain differences in susceptibility to autoimmune disorders between different ancestry groups. And that's partly motivated by the fact that we know that the incidence of many autoimmune disorders, it's significantly different between European American and African American individuals. This is kind of a summary that we put together a while back now, where you can, for example, see that psoriasis, it tends to be more prevalent in European ancestry individuals as compared to African Americans. And in contrast, for example, SLE shows the opposite pattern. We also have some evidence that natural selection might have contributed to those patterns. I'm going to go over this quicker. So the challenge with this type of analysis is that a lot of what we do is to just take these genome-wide association hits, so these genetic variants that were postulated to be associated with disease. But the challenge with that is that we still today, though there has been hundreds or thousands of variants associated with autoimmune disorders, we still have very little understanding about what are the actual causal variants and what are the genes that are associated with these diseases. So what we get out of these GWAS are something like shown in this cartoon, that is for every single SNP in the genome, we have an association with the disease. So you kind of get something like this, and anything that passes a certain threshold of significance here in the y-axis, it's called significant. And then how people link these GWAS hits with particular genes, it's often just based on location. So for example, imagine that you have IL-13 that is close to this particular hit. Most likely you will say that this GWAS hit is for IL-13. Yet there's actually another gene here that I made up a name. It's not a real thing. I just call it XZX whatever. And the point being that, because you don't really know much about this gene, you kind of tend to ignore it, right? But it could arguably be that these are the gene that is actually important for the disease. So one way to get around that is by doing what we call colocalization analysis, where you try to see if the signal that you observe for the GWAS hit is similar to the signal that you observe for the EQTL. In this particular case, for example, imagine that you see, you test like if these SNPs are associated with the expression of IL-13, and you see that there's no association. So there's no overlap in the signal. So it doesn't seem to be the case that the GWAS hit is explained by variation in the expression levels of IL-13. In contrast, when you do it with the other gene, you see that there's an overlapping signal. So in this particular case, you will say that this GWAS association is probably driven by variation in the expression levels of this gene. So we wanted to, because we have a lot of EQTL data in all these different cell types, we perform this type of analysis to try to narrow down to a set of genetic variants and genes that putatively are causal in a panel of, in this particular case, we used GWAS data from a total of 14 autoimmune and inflammatory disorders that are listed here. So this is not very important, but basically the point being that now we have a set of genes and regulatory variants that potentially are the mechanistic drivers of susceptibility to these diseases. And now we can start asking questions as, for example, are these genes that tend to be linked to susceptibility to this disease more likely to be differently expressed between the two populations? And the answer is yes. So
Louis,
these genes are much more likely.
You're over.
Yes, okay, sorry. I'm gonna finish.
You really have to finish. Thank you.
Yeah. Okay, so anyway, I think I covered most of it. So
Good.
I'm just going to finish by thanking the people that did the work. Most of the work was actually led by Haley, and of course all the funding agencies.
Okay, thank you. Okay. First of all, I'd like to again thank our sponsors for this session, Brooke and Duncan McLean and Karen and Eric Simon. And I'd like to get to the question and answer part if all the speakers would unmute and come on. Some of you may have answered some of these questions and answers. I'm going to pick a few that are relevant to celiac disease first, and then we'll go with some of the broader questions. The first is for Dr. Jabri. Does not your mouse model have overexpression of IL-15 in both epithelium and lamina propria? Are both necessary?
Yes, the answer is yes. And the reason why in this mouse model both are necessary is because this cytokine is expressed in a location-dependent manner. So when it's overexpressed in the epithelium, it's the reflection of a stress response in the epithelium, and then it's going to act to the cells nearby in the epithelium. When it's expressed in the lamina propria, it's going to act on antigen-presenting cells and change the nature of the immune response to gluten. So the reason it's required in both places is because it reflects the requirement for this interaction between the stress response in the epithelium and the adaptive immune response against gluten.
Okay, thank you. The next question is for Dr. Answer live. The next question is for Dr. Anderson. What is cross-reactivity of autoantigens, autoantibodies? We've seen patients with type 1 diabetes, autoimmune thyroid disease who have persistent elevated celiac serologies despite a gluten-free diet, but they normalize their duodenum or have mildly elevated serologies without evidence of celiac disease.
Yeah, this is a really interesting question about I guess the issue of cross-reactivity. Is there something going on, and how are these things interconnected? And it can get really complicated very quickly. But one simple thing I will say is, there is obviously a connection between type 1 diabetes and celiac disease, and we think most of that is probably driven by the HLA, which is the encodes the genes that are the ligands for T cells. And these Banna talked about it in her talk, right, about how there's this interesting property of DQ2 and DQ8 and the positive and negative charge. And it's probably the properties of these HLA molecules that allows the self-antigens or the gliadin antigens to bind promiscuously to promote autoimmunity. And then trying to connect it back to my talk is you can easily imagine in your mind that there might be a disconnect between what's happening in the thymus and what's happening in the immune periphery, right? So the thymus can present some antigens and get rid of T cells, but when there's this mismatch problem where the antigen binds to the HLA in the periphery in a different way, you can see how autoimmunity gets triggered. Now, I know that doesn't answer the cross-reactivity question, but where this cross-reactivity question really comes up more, I don't think there's cross-reactivity between thyroid antigens and type 1 diabetes antigens for the most part. I think that the connections of patients having those things is more related to HLA, but it still doesn't discount that there may be some infectious cross-reactivity. And so for that, despite that being a prominent hypothesis, we've really struggled in finding those connections. Is there some sort of environmental insult where you have cross-reactivity? And so I think the jury is still out on that question.
Okay, and there's a second question to Mark Anderson concerning gluten-specific T cells. Are these essentially determined to react to gluten by random rearrangements of TCR after birth that persist thereafter and render people the potential to develop celiac disease depending on the other factors? Or are these T cells evolutionarily predetermined to react to gluten? It's kind of existential.
Well, I like it. Very existential question, right? And it's probably the former rather than the latter, right? That you have to have, the way to think about it is you have to have the certain HLA type to be even at risk of getting celiac disease. And then there's random chance that happens in the thymus. Do you happen to randomly make a T cell receptor that can react against the modified gluten in the complex of that HLA molecule. And so this might explain, this gets back to what Gary and I were talking about, this identical twin stuff, right? If you had identical twins with celiac, sometimes you don't have concordance just because of random chance related to this event.
If I may add something, I think what is extremely important, and you see that in mouse models, any mouse with any MHC phenotype will respond to gluten. So it's because gluten has so many potential peptides. So it's not the capacity to respond or present gluten. Again, it goes back to the fundamental question of how do you amplify the immune response? What is the perfect storm that can lead to that? So I think this is an extremely important point, because be it in rheumatoid arthritis, be it in type 1 diabetes, in celiac disease, there are always very particular HLA molecules. And it is not that the antigen that Mark has presented cannot be presented by other MHC molecules. It's that there needs to be a combination of factor that enables this amplification of the immune response. And I think this is very important to keep in mind. So I think it's not just about the genetics and the frequency, it's about this combinatorial effect that needs to take place.
Yeah, hi, this is Gary. So I wanted to amplify again on and how important this notion of having predetermined versus a collection of stochastic events that contribute ultimately to the development of clinical disease. There's many people have evidence of autoimmune responses. Enormous number of people have, for example, anti-nuclear antibodies. And there is a burden of either epigenetic changes or environmental exposures that accumulate over time. And you need this perfect storm where everything has to be in alignment in order to actually get the disease. And if you have the correct HLAs and other things in advance, then it is easier to hit that critical mass. At least that's how we view it in the rheumatoid arthritis and lupus world.
Louis, anything about these factors regarding the microbiome or your talk in terms of antibody generation?
I mean, honestly, nothing specific that comes to mind.
Okay, very good.
But Louis, in what you have shown, right, where you see this slight genetic differences in immune responses, you can again think about how the combination of small variations in different genes can put somebody at higher risk of developing.
Yeah.
Okay, good. Okay, here's a question from Peter Green. Mark, dental enamel defects are very common in celiac disease. Have studies looked at the prevalence of antibodies to the protein in AIR patients been looked for in other patients with dental enamel defects?
So the quick answer is no. I mean, we really just unraveled this. We just published this a few months ago, and we're looking for collaborations. So if you see such patients that you're wondering about, we'd love to test them and see if this is true. We did, we did hear about a case at UCSF, or two cases at UCSF. There's siblings that had celiac and enamel hypoplasia, and we're working on trying to get the blood from them right now. So this is an exploratory area, but I wouldn't be shocked that it applies outside of this collection of rare patients with AIR mutations.
Okay, and let's get into a topic dear to our hearts, something about SARS-CoV-2 vaccines. How will RNA vaccines impact those with autoimmune disorders? This comes up a lot. It's a big clinical question. I don't know if you get them, Gary, on your rheumatoid patients, but the celiac people want to know about this. So who's going to take a shot at that one?
Well, this is Gary. So I think we don't really know yet right now. There are enough studies on, or at least observational studies on patients with lupus and rheumatoid arthritis that have been infected that suggests that A, the course of their disease is not particularly different from many others, and B, the medications that they're on not only don't seem to exacerbate infection, but also in some cases have been used in therapeutic trials like anti-IL-6 approaches and currently T-cell co-stimulation and TNF blocking approaches. So my suspicion is that the simple expression, well, not so simple, but the expression and immune response against the spike protein will provide protection without an abundance of untoward effects, because again, the much more complicated response to the actual virus, which is associated with antiphospholipid syndromes and clotting and all kinds of things, has not occurred to any great extent in the patients with autoimmune diseases, at least the ones that we see.
Okay. Louis, here's a question for you. Do your results suggest that there are different population-specific responses to bacteria versus viruses? What are the implications for COVID-19 given observed population differences?
Yeah, so I mean, at this point we can only speculate, but so one of the patterns that is the most striking from this recent data is this relationship between genetic ancestry and interferon response. And as Mark mentioned, type 1 interferon response seems to be a really important type of response for the severity of COVID-19. So it's probably going to be, I mean, of course, we don't know how these in vitro assays translate to in vivo responses, but we do know that initial interferon response is going to be very important, but also the interferon response later on, if it's not controlled, is going to be associated with the cytokine storms, right? So I think it's going to be a question of time and dose. We don't know, so we do not know if, like, among COVID patients of different genetic ancestries, that interferon response is going to be different, something that we are very interested in exploring in the future. But it does suggest that there's some differences there that could be potentially important.
Okay. And there's another question to you, or to Dr. Anderson. What does air intestinal failure teach us about celiac disease?
Well, I think the quick answer would be there are other kinds of intestinal failure besides celiac disease that it can have, a different target could come along. And, I think that the thing I was trying to get at is it was really mysterious in these APS1 patients why they were having this intestinal problem. We know that it's not celiac disease, but now I think we're getting onto a specificity. And so what I do wonder about is a patient's coming in where it's frustrating and they haven't really been clinched as a diagnosis of celiac disease, yet they are having intestinal problems. And we'd be very curious to see if there's, again, a connection between what we've been seeing in these patients with those kinds of patients.
And do pathologists look for the presence? I know they look for plasma cells and they comment on a lot of other cells. I don't see that they specifically look for enteroendocrine cells in the epithelium.
I think that
obvious to a pathologist?
Yeah, I think that's correct, but I think that you can't, that they are histologically detectable. You can do, and then if you have a hint that it's going on, you can do a special stain for the cells. Yeah.
And there's a question, another one for you, Dr. Anderson. Why are specific organs targeted in different patients with APS1?
Right, fantastic question, right? So a couple of quick things, right? So there's you saw on that slide, there's a pattern to this disease, right? And so something that's interesting about the disease, even though there's this fundamental problem in the thymus with making self-proteins, one of the features that you don't see in these patients is they don't get rheumatoid arthritis, they don't get lupus, right? And so it's teaching us something, right? The mechanisms that control tolerance for those diseases must be distinct. But then if you start talking about why there's some randomness between patients, that's also interesting, right? So if you can find siblings sometimes, right? They have the identical mutation in AIR, right? They've inherited 2 bad copies of AIR from mom and dad, right? So the AIR mutation is the same, yet they'll have different clinical features, right? And so we think that and Ivana brought up mice too, right? We can even more highly control it in mice, right? We can make mice where they're genetically pure and they still have the same mutation. And even there you see randomness. And we think that randomness is probably related again to how the T cells generate the receptors, and that's a component of why you see variation from case to case. And then there could be other relationships, again, getting back to what Vanna said, is in the target tissue, there's something going on. There's some sort of insult. And then that can get over a threshold and then promote the whole process from unraveling.
And one more question for Lewis here. There's two parts to it. I'm going to try to simplify it. Do your data suggest also a different clinical outcome when infected with flu?
Yeah.
Or does it depend on the lung immune response more than peripheral immune response?
I mean, it's hard to tell, but we do know that the incidence of flu, African ancestry individuals tend to have like twice the incidence of flu as compared to European ancestry individuals. Actually, the same type of racial disparities are observed today in COVID patients, as you all probably know. So it is possible, but again, our assays are very reductionist in the sense that we are really just infecting these cells in vitro. So it's unclear to which extent those similar differences in, for example, interferon response are going to propagate in vivo. So I will think that there's something there because we know that some of these differences clearly are genetically driven. So the hypothesis that there should be an impact, but we didn't test it yet.
Okay. Okay.
Can I just say
yes,
one thing?
Absolutely.
I think one of the really interesting phenomenon that we are seeing when we compare population, and this is something Luis and I are very interested in, is that the MHC in African Americans, the predisposing celiac MHC is similar. And African Americans have a high tendency to develop type 1 diabetes, but you almost have no celiac disease. And this to us is incredibly intriguing and suggests that there might be some genetic factors that somehow protect them from celiac disease by still predisposing them to type 1 diabetes. So I think that studies that would compare and try to understand those differences can really teach us also about protective mechanisms.
Okay, I think our session is over. I certainly learned a great deal, and I'm happy I attended this session. And again, thanks to our sponsors, our generous sponsors who allow us to put on this very high quality session. Thank you all.
Thank you. Bye.
Thank you.